Good morning. My name is Lucia at the University of Rhode Island, and the title of this talk is Modeling Ischemic Livers Using Nash Equilibrium. My co-authors are androulakis at Rutgers University and Mustafa Korkut Uygun at Massachusetts General Hospital. The outline of this talk is given on this second slide. I will first introduce you to liver ischemia, liver perfusion and reperfusion, and the motivation for this work. Next, I'll give you a summary of the Nash equilibrium modeling and simulation approach. Nash equilibrium modeling and simulation is a first principles approach in which enzymes are players in a multiplayer game. Then I will present a metabolic model of the liver, which includes all of the key pathways in any cell. The main focus of this work is on numerical simulations in which we concentrate on ATP content and energy recovery during perfusion. I'll then compare the numerical results with experimental data that we've gathered, and finally make some conclusions about this work. The motivation for this work stems from the fact that there are approximately 10,000 new liver transplant patients per year in the United States, but only 7,000 transplantable livers are available. Unfortunately, transplantation is the only solution to liver failure. However, there are 6,000 or approximately 6,000 ischemic livers that are discarded each year that could possibly be made transplant ready using machine perfusion. Liver ischemia is simply low blood flow through the liver. This in turn can cause low oxygen levels or hypoxia, which can damage cells. Reperfusion is just the mechanical pumping of blood through the liver blood vessels. In addition, nutrients and pharmaceutical additives are often used to help restoration. However, this is not without problems. Liver reperfusion can cause injury due to inflammation and oxidative stress. On the right is an illustration of a liver perfusion machine. As I mentioned earlier, the Nash equilibrium approach to metabolic modeling is a first principles approach that's formulated as a chemical equilibrium problem. The key ideas behind the approach are as follows. 1. Enzymes are players in a multiplayer game. And what this means is that each enzyme minimizes the Gibbs free energy of the reaction it catalyzes, subject to a set of constraints, which include atom balances, the nutrient pool, feedbacks, regulation, and so on. 2. The goal of the metabolic network, on the other hand, is to find the best overall solution, subject to the natural competition for nutrients among the enzymes. Determining a Nash equilibrium point requires the solution of n nonlinear programming subproblems. The interesting thing about this is that the solution that's found is not necessarily the best solution for the individual enzymes. And of course, everything depends on the initial state of the system, which is defined in terms of the nutrients available. Flux balance analysis, or FBA, has been the mainstay for determining steady-state fluxes throughout a metabolic network for many years, and it has many variants. Unfortunately, it also has some disadvantages. 1. It doesn't include chemical intermediates. 2. It doesn't consider charge balancing. And 3. Enzymes are often not included in the model. On the other hand, the Nash equilibrium approach takes into account the generation and/or consumption of chemical intermediates. It uses charge balance reactions. And if enzymes are important, enzyme-substrate reactions can be included in the model. On the right, I show the charge balance reaction for glycolysis. This is an overall reaction in which many of the chemical intermediates in glycolysis are not included. Many textbooks in biology or pharmacy state that this is interpreted as 1 mole of glucose gives 2 moles of pyruvate. In the figure below, you can see that some of the chemical intermediates, for example 3PG, are generated, while others, like G6P, are consumed. However, in flux balance analysis, these intermediate species are not included, and therefore one winds up with an incorrect interpretation of the overall reaction. In fact, it is easy to show that 1 mole of glucose does not go to 2 moles of pyruvate because of the accumulation and/or depletion of chemical intermediates. The importance of correct charge balancing cannot be overstated. Incorrect charge balances lead to incorrect mass balances, which in turn can lead to algorithmic failure, and this is generally due to an incorrect number of hydrogens on the chemical species involved in the reaction. In addition, not all atom balances are linearly independent. In fact, this is a pervasive problem in metabolic networks. There are large numbers of dependent constraints. Therefore, you have to find a way of correctly formulating the problem, and the way to do that is to keep only those species or mass balances that correspond to the transfer of elements. On the right, I give an example. Note that the reaction is charge balanced. Note also that the only elements that are transferred are hydrogen and oxygen. However, there are 5 element balances. But because only hydrogen and oxygen are transferred, those are the only 2 element balances that are linearly independent, and those are the only 2 that should be kept in the model. The metabolic network model for the liver used in this study is shown on the right. Note that it includes all of the common pathways in any cell. Pathways are shown in red, metabolites and cofactors are shown in black. The blue dashed lines indicate inter-pathway transport, and the green lines indicate the fatty acid switch between fatty acid synthesis and beta oxidation. The numerical simulations important to liver ischemia and machine perfusion are static cold storage and energy recovery during machine perfusion. In static cold storage, livers are stored at 4 degrees C, and as a consequence, the ATP content of the liver depletes over time. The goal of energy recovery during machine perfusion is to increase ATP generation, which in turn will increase ATP content and energy charge. ATP content is important because it's been correlated to liver viability for liver transplantation. The figures on the right show a comparison between experimental data and Nash equilibrium simulations for static cold storage of rat livers. The details of the experiments and the numerical simulations are briefly described on the slide. The most important thing to glean from this slide is the fact that the Nash equilibrium simulations correctly capture the experimentally observed time-dependent behavior of static cold storage of rat livers. Some of the key details associated with the static cold storage simulations include the fact that the liver received no nutrients or oxygen, so there was no ATP generation, and ATP depletion was the result of the anabolic pathways in the model, which are listed below. Other key observations include the fact that the concentration of ATP over ADP fell below the normal range, and the energy charge was also low. The next set of numerical experiments that we did were related to energy recovery during machine perfusion. The key points associated with these simulations were as follows. 1. The initial state of the machine perfusion simulations was taken as the steady state solution for the static cold storage simulations, the only difference being the temperature was raised from 4C to 21C. 2. Nutrients were added. In particular, we used Williams medium E, which consisted largely of glucose and various amino acids. This drives glycolysis, which will produce a small amount of ATP and NADH. Glycolysis, in turn, produces pyruvate, which then produces acetyl-CoA, and this drives the Krebs cycle, which generates the bulk of ATP, NADH, and FADH2. 3. Oxygen was added. This drives oxidative phosphorylation, which produces ATP. On the right, I show once again a perfusion machine. A sample of the machine perfusion simulations we conducted are shown on the right. Note that ATP content and the ratio of ATP to ADP increase with glucose addition and with increasing oxygen. However, note that both curves appear to have asymptotes at very high oxygen flux. Some of the key details associated with the machine perfusion simulations are shown on this slide and are as follows. 1. The liver received nutrients from Williams medium E and various amounts of oxygen. Williams medium E was normalized to 1 picomole of glucose per cell, and oxygen flux varied from 0 to 30 picomoles per minute per cell. 2. ATP was generated from the catabolic pathways in the model, which include glycolysis, the Krebs cycle, and oxidative phosphorylation. Other key observations include an increase in energy charge, an increase in pH, and a measure which is shown at the bottom of this slide, which fell within the normal range for energy state of the cell. The main conclusions of this work are shown on this slide and are as follows. 1. We proposed a metabolic network model for the liver, which included all key pathways in any cell, compartmentalization, and interpathway transport. 2. We performed 2 sets of numerical experiments using the Nash equilibrium framework. The first set of experiments was concerned with ATP depletion in static cold storage. The second set of numerical experiments was concerned with ATP generation during machine perfusion. 3. The main results of these simulations show that the Nash equilibrium approach can successfully simulate or capture the qualitative trends of both ATP depletion in static cold storage and ATP generation during machine perfusion.